That was very slow. Okay, guys. So, how's the studying coming along? I hope you're doing some sort of studying, because Monday, a whole bunch of you are taking an exam. Which reminds me, I need to write an exam. Okay. I don't want to write it, you don't want to take it, I don't want to grade it. Can't we just talk about how we love science and all get A's? I wish we could, but people frown upon that. What was I going to say? Oh, 2 brave souls have requested the oral exam. So if you're considering the oral exam, please let me know, before the end of the weekend, because you will be taking it the same day you would take your normal exam, but during my office hours. So, but please let me know, because I need to plan accordingly. Okay? For the Gene Expression Project, most everyone I've seen so far has their 3 aims. Let's make sure we have our 3 aims. If you're still struggling, you need to communicate with me, because, well, that helps, hopefully. Exam frequency. So here's your list of stuff you need to focus on. Always, anytime you can compare and contrast, you should be comparing and contrasting. So we did a fair amount of that in the beginning with DNA and RNA, and DNA and RNA polymerase. We've talked about the transcription, the actual mechanics of transcription in bacteria and eukaryotic cells. We talked about the concept of coupling transcription to processing and the importance of the C-terminal domain. We talked about regulatory mechanisms and the basic ingredients, DNA elements, transcriptional regulators, promoters, enhancers, locus control regions, insulators. We talked about how these regulators bind to DNA, and now we've been talking about regulation of gene expression, what are activators, what are repressors, how genes are organized into operons. And we're finishing up with talking about regulation of initiation, elongation, and termination. A lot of what we talked about, and a lot of what you think about when we talk about regulation of transcription, is regulation of initiation, right? Binding of an activator, recruiting general transcription factors, starting transcription. But you can also regulate elongation and termination. And then there's further regulation after you have your RNA that's still going to affect gene expression. Oh, I forgot my class list. Hopefully I know everyone. Okay, so regulation of initiation, this is what we're talking about. Does this happen? Do your general transcription factors or mediator get recruited so that RNA polymerase could actually start transcription? So I'm going to take a moment to kind of back up and remember that gene expression is usually activated or repressed, actually, by signaling through a receptor that originates at a receptor, right? Because some sort of extracellular molecule is going to bind to the receptor, and then a whole bunch of magic's going to happen involving the cytoplasmic tail of the receptor and phosphorylation and recruitment of kinases and phosphorylation of other things. And then ultimately the entry of some sort of transcription factor to the nucleus, where it binds to an enhancer region and recruits general transcription factors, mediator, whatever needs to happen, and gene expression. So there's your big picture. I know it's easy to kind of get tunnel vision. Yes?
What is the time that it will take?
How long does this take? Depends on the pathway. Depends on the pathway. It really depends. It really does. Every signaling pathway is different. The requirements for how many transcriptional activators you need to assemble, that's different. I think that, so for example, in the past I've studied the NF-kappa B signaling pathway. And I've used as a control treating my cells with TNF-alpha. So there's a signaling molecule that's going to bind to a receptor and activate gene expression. And I looked at NF-kappa B activation through a reporter assay. And I've been able to see very high levels of gene expression in under 24 hours. Now, mind you, I'm sure it's way faster. It's just that I checked at 24 hours or a little bit less. I think I've checked in as little as 12 hours. I believe that signaling like that, something that's very strong like TNF-alpha, is going to very rapidly activate gene expression. Let me see, let me give you another example that I've seen in research. I did some work where we did a time course. Oh, it was viral gene expression, which is different because their promoters are fairly strong. But we looked at gene expression of a viral promoter and saw that within, at the RNA level, within something like 2 hours. So depending on the gene and the strength of the promoter, things can happen fairly fast. Which makes sense, right? Usually you're responding to something, right? And then as we see as we move forward, some promoters are actually kind of everything sitting there and it's stuck at that abortive initiation phase. And you need to kind of release that and allow, so it's actually everything's poised and ready to go. And that's going to be extremely rapid. So we'll see that in a minute. Okay. So here's your big picture. I want you to constantly keep this in mind as we go through all of this. Okay, so let's talk a little bit more about initiation, because that's really what we've been focusing the most on. And you can think of these gene regulatory proteins as being regulated in a variety of ways, right? So the thing is, it needs to come and bind to the DNA in order to activate gene expression. But what's gonna regulate whether that happens or not? What's gonna control whether it can bind, right? So normally you can think you've got protein synthesis, which results in some kind of gene regulatory protein that then binds to DNA and gene expression happens, right? That's the simplest way of thinking about it. But then think about all the regulation that's going to control whether or not it can bind, right? You can have a ligand that binds and creates an active form of your activator. You can, or repressor, your gene regulatory protein. You can have a covalent modification. Perhaps you need phosphorylation to activate. There could be some kind of dimerization. So a second protein might need to bind to allow. We see that with the JAK-STAT pathway. Those of you who remember studying that. Sometimes you need dimerization to allow activation. Sometimes the gene regulatory protein's always there, but it's inactive because it's bound to an inhibitor. Who can tell me an example of a gene regulatory protein that's always there, but it's normally bound to inhibitor? We need to get rid of the inhibitor through phosphorylation and degradation. Yep.
Is it E2F?
Oh, well, E2F is another one that I wasn't thinking about, right? E2F and RB, right? Okay, and I just heard NF-kappa B through here. So that's two examples that you guys were able to think of things we've heard about in the past that normally are there and there's an inhibitor. And so that kind of gene expression is going to happen fairly rapidly because you don't need to upregulate the expression of your activator. You just need to kind of activate it by getting rid of your inhibitor. And it's easy to phosphorylate or to degrade something, right? It's easier than transcription translation. You could also have regulation of nuclear entry, right? So sometimes the gene regulatory protein, and this is an example, this is NF-kappa B and JAK-STAT, are both cytoplasmic, and upon stimulation of gene expression through you're activating a signaling pathway, the transcription factors enter the nucleus, right? They dimerize and enter the nucleus, or inhibitors on it enter the nucleus. So whether or not your nuclear cytoplasmic can regulate your activity if you're a gene regulatory protein. And then there are some gene regulatory proteins which we actually haven't talked about at all in this class, but are normally bound to the plasma membrane. And then there's either cleavage, usually cleavage that results in release from the membrane-bound form, and then it can traffic into the nucleus. But really, so this looks like a whole lot of stuff, but what you need to ask yourself is it available to bind to the DNA or not, right? If it's in the cytoplasm, can it bind to DNA if you have a nucleus? If you're bacteria, that's an irrelevant question. But if you're a eukaryotic cell and you're in the cytoplasm, can you bind to DNA? Now, who thinks you can bind to DNA in the cytoplasm? I guess to understand this, you need to realize that there's regulation whether or not proteins go in and out of the nucleus. They have nuclear localization signals that allow them to be transported into the nucleus. And so if you don't have an NLS, nuclear localization signal, or it's not exposed, you're going to be stuck in the cytoplasm. So think back to 408 for those who've taken it, and just kind of appreciate that for those who haven't. Ligand binding, that's a lot about changing conformation. We talked about that with the TRP repressor, right? Where tryptophan binds and changes the conformation and allows binding to that major groove of the DNA, right? Covalent modification, same, you're changing a binding site. Dimerization, that also often is about allowing something to get into the nucleus, but might also be creating a binding site that requires 2 subunits. Okay? Any questions about this? This is a really nice summary slide. This is why I love Alberts and do not love the other textbooks, because they go through lots and lots of specific examples, of which we're going to go through some of, but this is really basic concept, right?
What's up with the big black shapes?
I don't know, it's weird, huh? What's up with the big black shapes? I think what it's trying to tell you here is this is something's maybe phosphorylating and getting rid of, I don't know, it's weird, right? Don't worry about that. You can get the point, we're good. Okay, is anyone looking at this and it seems like it makes no sense? Please raise your hand, because I would like to expand on this, because these are major concepts that need to be understood for you to survive this exam. So if you're like, oh my god, what is she saying? Yes?
So it's just kind of showing that those are like different ways that it could be regulated.
Yep, just many mechanisms to regulate whether or not a gene regulatory protein, repressor or activator, can do what it needs to do, which is pretty much bind to DNA and activate or bind to DNA and repress. So you're either changing the shape, excluding it from the nucleus, stuff like that. It's mostly shape changing and exclusion from nucleus and covering up binding sites, right? I think that's the most of it. Okay? I'm going to move on. Okay, here we go. Okay, so you saw this a few times. I just always want to put the fruit back up. Just the concept that is fairly complex, and one gene might be regulated by multiple gene regulatory proteins. I show this as a very simple thing. Gene regulatory binds, right? Multiple gene regulatory proteins are going to bind to multiple DNA elements within a control region that might be as much as 100,000 base pairs long. So we're talking about insane levels of regulation, and so you'll see this as we go through some of these examples. Actually, I think I have an interferon example. Okay, and what I mean by control region, this is a region of DNA that will bind to things and control whether or not gene expression is going to happen. Okay, so here, let's go through some examples of regulation of initiation. So there's this one gene regulatory protein called ELK1, and it activates transcription in response to mitogens through direct recruitment of mediator and HAT. So mitogens are extracellular signaling molecules, something that might be secreted by another cell that's going to activate, it's basically going to push a cell in the cell cycle, and it's going to promote mitosis. So whenever you talk about cell proliferation, there's usually some kind of mitogen involved. You might have seen that a lot in the CMV gene expression project. Blah, blah, blah activates cell proliferation, maybe. You would need that in the immune response. And so what happens, so ELK1 is a gene regulatory protein. So automatically you should think it has a DNA binding domain and it has an activation domain, right? And so the DNA binding domain is binding to DNA. This serum response factor is just involved in the binding. Don't worry too much about it. And normally it will bind, but it's not able to activate gene expression. And if you have signaling, so a mitogen binds to a receptor and signal transduction happens, then what will happen is you'll get activation of a MAPK, which will then phosphorylate the activation domain of ELK1, which allows it to then recruit and interact with mediator and HAT complex. So what's a HAT going to do? Acetylate, so you can see like, oh, right, that promotes gene expression. Mediator, you know, that's involved with the recruitment of general transcription factors. Those are both things that should activate gene expression, and that's what it's supposed to be doing, right? It's activating transcription in response to basically growth factors, okay? So that's one example of regulation of initiation, right? Because we're recruiting HAT, mediator, and then all the other things needed for RNA polymerase to be recruited. Okay? So here's another example. We're going to go back to GAL4. We talked about that briefly when I introduced the concept of DNA binding domains and activation domains, and that's what's used in these two hybrids. So GAL4 is actually a transcription factor that regulates genes involved in galactose metabolism. So it's not only used in these two hybrids, it's actually there's a real expression system in yeast. So in the absence of galactose, GAL4 activation domain is blocked, right? Because it's regulating expression of genes required for galactose metabolism. If there's no galactose, it should be off, right? Do you want to metabolize galactose when galactose is not there? No, that seems silly, right? So here's the DNA binding site for GAL4. Don't worry about what it's called. And so normally when there's no galactose, this GAL80 is binding and preventing activation, okay? And so when galactose is present, then there's actually, and this is a unique mechanism, but it's kind of cool, then GAL3 is going to come and it's going to bind to GAL80, and it binds more strongly, so it's going to remove it from GAL4, and so now GAL4 is available to interact with these other proteins, one of which is mediator, and another is SAGA, and basically this is a complex of proteins that recruits title-binding protein and the general transcription factors and activates gene expression. So the last one was an example of phosphorylation, which results in recruitment. And this is an example of an inhibitor that's removed. And so these directly relate, to this, right? Okay, so you get gene expression. Yes?
So this one would be an example of unmasking?
Unmasking, because it's present in the nucleus, it's there, but it can't, the activation domain is covered up.
I know previously that would be reminding binding.
That would be phosphorylation.
Okay.
So you get phosphorylation of the activation domain, which allows recruitment of the other proteins, co-activators, mediator. Okay, so here is an interferon beta gene, which you guys have probably thought about a fair amount in your projects. So regulation of interferon beta involves cooperative binding of many different regulatory proteins. And so there's an interferon beta enhancer that needs to be bound by a whole bunch of proteins, and some should be familiar to you. You've got NF-kappa B, you've got P50, P65. You've got these IRFs, interferon response factors, right? And then you've got JUN and ATF, right? So the gene is actually off unless all of these genes are upregulated and can enter the nucleus and bind to the DNA and activate gene expression. And so this is actually how they look sitting on the DNA. So there's your DNA. And so JUN sometimes works with FOS. You guys may have seen that before. It also can dimerize with ATF too. And it's this coiled club. Remember, we learned about this is how these different transcription factors bind to DNA. This coiled coil structure that kind of sits right on the major groove there. Then you've got your IRFs and P65 and P50, and they all kind of cluster in this enhanceosome, and all together can act in recruitment of general transcription factors and RNA polymerase. Okay? Lots of stuff. So you can imagine that this is tightly regulated. If for some reason IRF3 is not upregulated, it's not going to be available to come bind to the DNA, and interferon beta is not going to be turned on. Right? So if you were a virus and you wanted to downregulate any of these, it could have huge effects on the antiviral response. Right? Maybe? Maybe not? I see someone nodding. Someone's awake. So this is pretty stringent regulation, right? Okay. Are we happy today? It's Friday. It's supposed to be like 70-something degrees later. Yeah. Very exciting. Okay. Okay, so gene expression is constantly changing in response to cellular conditions. So I just gave you a few examples, right? Who can give me other examples of what might affect gene expression? Heat shock. Okay, thank you for introducing what I'm about to talk about. Heat shock is one. Right. Okay. Anyone want to add to that? Is anything that makes the cell unhappy, or anything that the cell needs to respond to, or anything that...
Pretty much any change is going to want to regulate how it's acting.
Any change, absolutely. Nutrition, environment. There are pathways for sensing metabolites, so you know if the environment is favorable to go into cell cycle, right? Especially if you're bacteria. Okay. Gene expression is affected by binding of hormones to steroid hormone receptors, right? So you guys have heard of hormones. Have you heard of hormones before? Who hasn't heard of hormones? Okay, just making sure we're all awake. Right, so you can actually have a ligand, which would be your hormone, and hormones actually diffuse right through the plasma membrane, right through the nucleus, and go right in and activate gene expression. You could also have gene expression induced by binding of signaling molecules to receptors. Right, this is an NF-kappa B pathway, so this could be your TNF alpha, which results in blah, 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 things binding to DNA. Right. I could go through a whole signaling pathway, but I won't do that to you. You all know this one by now, right? A lot of you guys should know this one by now. Okay, so then there's, right, so we talked about regulation of initiation, right? So then you can regulate at the level of elongation. This takes advantage of early stalling, which is also called promoter proximal pausing or abortive initiation. Remember when you start transcription, you get like these 10, 20 nucleotides transcribed, and then there's the stalling. So that could be a point where you can actually regulate and keep this assembled transcriptional unit waiting to be released from this abortive initiation. So you're poised for rapid transcription. And so heat shock, thank you for the introduction, heat shock works this way. HSP70 is a chaperone that plays a protective role in response to heat shock. So self-stress in general, don't just think about it as high temperature. There's a lot of self-stresses that will activate HSP 70, and it prevents, does many things, but one of the main things it does is prevent aggregation of partially synthesized or misfolded proteins. So sometimes if there's an excess of misfolded proteins, that in itself will activate the heat shock response. So the way it works is it converts this stalled RNA polymerase into an elongating complex. So normally you've got, this is how your gene looks, okay? So you've got some kind of element here, and you've got your DNA wrapped around your histones, and then this is actually a chromatin nucleosome remodeling complex that's going to remodel the nucleosomes, and heat shock factor is going to bind to a heat shock element, and recruit RNA polymerase, and it's just kind of hanging out there in this stalled way. Let me take that back, actually. Heat shock factor cannot bind in the absence of heat shock. It needs to be trimerized. So normally you've got this kind of this transcription factor actually results in this stalled RNA polymerase just hanging out. And when you have heat shock, then you get trimerization. So you get a conformational change in heat shock factor to form this trimer, meaning 3 heat shock factors binding together. And only that will allow interaction with the heat shock element. That results in recruitment of mediator, and then you end up with conversion into the elongating RNA polymerase. Okay? So normally chromatin's kept open, RNA polymerase can bind and begin transcription, but it's at this stalled state. And the rise in temperature is what causes trimerization of heat shock factor. Binding to heat shock element recruits mediators and kinases to phosphorylate the C-terminal domain, because remember that's involved in elongation, and then you get elongation. That's an example, just one example, of regulation of gene expression, elongation, of the elongation point of transcription. Okay, so we talked a little bit about regulation and initiation, a little bit about elongation. We could also have regulation of termination, right? And so I'm going to just talk about the concept of attenuation. Who brought up attenuation the other day? Was that you? No. I think it was someone sitting over here. Was it you? No. Who brought up attenuation the other day? You? Yeah. And I remember being like, which attenuation are you talking about? We were talking about this attenuation probably, right? So where you're controlling the structure of the RNA and how that is going to either allow transcription to continue or result in a termination hairpin, right? That's what we're going to talk about. So that's one example of attenuation. One example. So this is where you're controlling the effect of the terminator sequence. And it's used to control, it's frequently used to control transcription of enzymes involved in amino acid biosynthesis. So we're going to talk about the trip operon here. And the effects are based on alternative RNA secondary structures. So this is in bacteria, okay? So usually, so the trip operon, it's an operon, meaning there are multiple genes transcribed. It's a polycistronic method, right? So the way it works is normally, so you've got this leader sequence that contains many codons for tryptophan, right? And if you've got low tryptophan, okay, and the other concept to understand is that often when you make, especially in bacteria where they're in the same place, once you make your RNA, ribosomes hop on and start translating, right? So in low tryptophan, what happens is you start transcription and you go through this leader sequence that contains these trip codons. If you have low tryptophan, then the ribosome's gonna stall because it's waiting for that tRNA to come in that has a tryptophan amino acid. And so it actually obscures this sequence here, which results in this hairpin forming, and that prevents this terminator hairpin. And this is the attenuator. So if you cover up this, so the thing to understand here is you've got 1, 2, 3, and 4. These 4 structures that can form alternative hairpins. And if you stall your ribosome, it covers up number 1 and results in 2, 3 forming. And then you can get transcription all the way through the operon, resulting in an expression of all the genes needed for tryptophan biosynthesis. And in high tryptophan, your ribosome's not going to stall because there's a tRNA just hanging out waiting to be added. And it goes through, and that kind of covers up 1 and 2 and results in this attenuator forming, because you can't have this 2, 3 form because the ribosome's covering it. And 2, 3, 4, 3, 4 hairpin forms, and that's your attenuator and stops transcription. So you never make it all the way through the operon. So it's the presence of these tryptophan codons in the leader sequence that are going to control this. And it takes advantage of the fact that the ribosome is going to hop right onto the RNA and start translating. Okay? So it's all dependent on the rate of translation. So we've been talking a lot about transcription, but here we're depending on the rate of translation. Now, why can't this occur in eukaryotic cells?
Translation, transcription.
Okay, where does transcription occur in eukaryotic cells? How about Vito? In the nucleus. Where does translation occur? Justine? In the cytoplasm, right. So this could never happen where you have, well, first of all, this is controlling operon, so that's already not going to happen in eukaryotic cells. But you'll never have your ribosome jumping onto your RNA before it's done being transcribed. Because remember, you have transcription, processing, export from nucleus. That's a fantastic, easy, tricky exam question, isn't it? Yeah, so low tryptophan, ribosome stalling, prevents this 3-4 hairpin because it allows the 2-3 to form. And so you would think that this 2-3 hairpin would attenuate, but it actually doesn't. So it's obvious for you to think to yourself, well, why isn't 2-3 stopping transcription? It's not. It's some other hairpin that I guess is easily passed through. Maybe it's weaker. I don't know the exact mechanism, but this is not stopping. Oops. This hairpin is not stopping gene expression. This is your attenuator. This one stops gene expression and kicks off RNA polymerase. This one allows RNA polymerase to pass through and transcribe the whole operon, which contain all the genes for tryptophan biosynthesis. And it's logical, right? Low tryptophan, you want transcription. High tryptophan, you don't. Okay? Any questions about this? It usually generates a lot of questions. Maybe. Okay. Yes?
Okay, so I just wonder, what's the purpose of the presence of attenuators? Because don't we have repressors for trip operon already?
Thank you. I like that. What's the purpose of an attenuator? Don't you have repressors for a trip operon? Well, often there are multiple mechanisms of regulation, and this is just an example of multiple mechanisms of regulation, both of which, oh, that's another really great exam question, isn't it? 2 mechanisms for regulation of the trip operon, right? 2 very different mechanisms, both which depend on the sensing the levels of tryptophan in the cell, right? Because the repressor binds to tryptophan, right? And so in the presence of tryptophan, it will bind to the repressor and bind strongly and prevent gene expression. And here, you're sensing tryptophan by whether or not tryptophan is being incorporated into this leader peptide by your ribosome. Oh, I love it. That's a good one. Who loves it? Okay. Yeah, you love it? Huh? Did you want an answer? That was the answer. 16.
So, I'm sorry. So with the presence of tryptophan, so do both mechanisms occur to prevent transcription or just one of them?
Well, I guess you can say that they're kind of sequential mechanisms, right? Okay, the question was, do both occur or just one or the other? So, regulation of initiation is going to happen before regulation of termination, right? So you can imagine that if you have high levels of tryptophan, your repressor is going to be bound, and this is never even going to start, right? But let's say it started because there were low levels, right? So there were low levels of tryptophan. Repressor didn't need to bind because you needed gene expression, but then tryptophan gets high in the cell, right? And so there's got to be some way to start to shut this off, right? So you can start to sense it this way and then create the attenuated structure. And then you're also going to have tryptophan that then can bind to the repressor and prevent further recruitment of RNA polymerase. So I think it's a combination. You have to have multiple mechanisms of regulation because it's not just does it start or not. It's also something that's happening that then needs to be slowed down and stopped. Does anyone want to add to that?
It's also not like high levels and low levels. There's a whole spectrum. So there can be low levels of transcription, and this is just another level of regulation.
Okay, great. So there could be low levels of transcription. Yep. So it's not just like with science. It's not just black or white. It's not just on or off, right? Okay. Anyone have any questions about this? That was a great question. Yes.
Question about operons. I've read that there are some eukaryotes that do have operons.
There are some eukaryotes that do have operons. Okay. And we kind of did talk a little bit about the locus control regions as a way to regulate a whole cluster of genes.
So an example of this question is states like are operons not eukaryotes?
I might word it as operons are primarily found. Yeah. If on an exam I said something that was wrong, if you within an appropriate amount of time brought me a well-reasoned referenced argument, then the teacher's not always right. Okay. Any other thoughts about this? And we'll move on. Okay. Transcriptional silencing. So we talked about regulation of initiation, regulation of elongation. I just gave you examples. Right? There are many ways these things can happen, but I didn't want to kill you with 30 mechanisms. And then regulation of termination, and then there's transcriptional silencing. Transcriptional silencing means the RNA is already made. You can still silence, right, by getting rid of your RNA, pretty much. Well, two things. One is through chromatin structure, and the other is through small RNAs. And there's even more than that, but those are the two I'm introducing to you. So in yeast, SIR proteins, they're silencing information regulators. SIR proteins establish and maintain heterochromatin. And so they're actually binding sites that these other proteins bind to and recruit these SIR proteins and result in recruitment of SIR2, which is HDAC, deacetylase, right? And so that's the first step in establishing repressive heterochromatin is deacetylation. And you can imagine future steps might be recruitment of what? Methyltransferase, right? Because you have low acetylation and high methylation, usually in repressive heterochromatin, right? So that's one way of accomplishing transcriptional silencing, keeping transcription off. Another is RNAi, which is a mechanism for targeting specific RNAs for degradation. And this happens primarily in the cytoplasm. So you'll have double-stranded RNA, specific siRNA, microRNA genes that are recognized and processed by a complex of enzymes into these RNAs that are complementary to the mRNA that's going to be degraded. And so it targets certain RNAs, sequence-specific targeting of RNAs for degradation. Viruses love to do this. And then I mean, there's a ton of microRNAs in our cell, actually in our genome. There's actually a huge amount of research going on looking at regulation of microRNAs in cancer. Because if you have, just like if you have upregulation of the wrong gene, it can result in cancer. If you have up or down regulation of the wrong microRNA, it can result in cancer because it will affect expression of certain genes. So this is, I don't know if that looks very nice, but so this is just kind of how it works, right? So you'll have your transcription of your microRNA by POL2, and it makes this kind of hairpin structure, and then you get processing of that structure into a microRNA, a pre-microRNA, and it's exported out of the nucleus, and it'll either interact with all these processing enzymes and target base pair with the mRNA and target it for cleavage, so literally cleave that RNA, or it might also bind to and repress translation. So that's another function. So those are two more ways. And I'm very sad to say, I was worried we didn't have enough time. I probably could have introduced more, but maybe we'll have a discussion for a minute. What are some other ways that we've talked about of regulating gene expression, right? So we've talked about regulating it from the perspective of, can RNA polymerase make an RNA, right? We've talked about it from that aspect. We've talked about it from the aspect of, can that RNA survive long enough to be translated into a protein, right? That's right there. So what other ways can gene expression be regulated that we've talked about? Gene expression regulation. So gene expression is, I guess, resulting in a functional protein. What might affect whether or not you get functional protein, or affect the function of the protein, or affect what proteins are made, what they look like? Yeah. Well, what, like, okay, so environment, but not like in terms of the outside of the cell and how it affects it, but what mechanisms are going to have an effect on going from gene? Because what we talked about is really reading the genome. How is the genome read and made into a functional protein that's going to do something? And a lot of this regulation is talking about whether or not you get the RNA and whether or not it can be made into protein. But there's a whole lot of other things that can happen that are going to affect how the genome is read and made into a functional protein. So what do we talk about? Yep.
Activators or repressors in DNA.
Okay, so but that's still, activators or repressors, that's still talking about whether an RNA is made or not, right? So let's talk about, outside of whether the RNA is made or not, let's talk about what kind of RNA is made, what kind of protein is made, what does the protein do? Yes?
So how about
Post-translational modifications, right? So that's going to affect, so right here we've talked about getting the RNA, and it's going to be translated, but then how it's modified is going to regulate what it does and where it goes. So that's, you've read your genome, you've made your protein, but your function is completely regulated by whether or not you're phosphorylated, you're sumoylated, you're ubiquitinated, a million things. What else? Yes?
I don't know, but wouldn't natural selection mutations cause like a frame
Okay, so you can get mutations that have a selective advantage that will result in a different protein, but that's in your genome. So that's, yeah, okay. So what else? I'm looking for specific things that we've mentioned, we've talked about. Maybe that's to do with, yes? Oh, that's one thing I was looking for, alternative splicing, right? So you've got a gene, but how it's processed is going to affect what RNA and what protein you're gonna get, right? What?
No, I'm just confused about the question.
Okay, sometimes I have a hard time getting my point across. Yeah, the question is really, what other things can be regulated that are gonna result in what your gene is turning into multiple different types of proteins. So sometimes it's whether or not your RNA is even made or not. But then there could be alternative splicing, there could be some kind of mutations that will change things up. What else?
Do you mean like signaling pathways? Problems in the signaling pathway?
No, I'm not looking for things going wrong. I'm looking for things that are going to affect. So one thing is RNA editing, right? So you can have editing of your RNA to then create premature stop codons, which will truncate proteins. Whether or not then things that control whether or not your RNA is ever going to make it out of the nucleus. All of those processing events all can be regulated, right? Were you raising your hand?
I mean, you can also regulate how much of the protein is made if it needs to trimerize or dimerize,
You can regulate, okay, so you can regulate how much your protein is being made.
or other proteins.
Yep. You can regulate levels of proteins that might need to heterodimerize with your protein in order to be functional. Yep.
What about regulating the axis of the gene to the protein?
Regulating the access of the gene to the protein. So you mean keeping something silent so that an activator cannot bind it? Yep. Yep.
What about where it's going when it's like EPR, little gene?
Right, so regulating localization, which has to do with whether or not you have signal peptides and stuff like that, signal sequences, but also have to do with post-translational modifications once you're actually made. Where will you go? Or what will you associate with? I just want you guys to think very globally here. It's not just recruit RNA polymerase gene expression. There's so much more that can happen. What about degrading proteins, right? If you degrade a protein, it's not going to function. A lot of proteins are degraded to turn things off. We learned about that a lot with cyclin. Okay, I guess you guys can go.